Dear viewers, greetings. In this video, we are going to see about analytical centrifugation. The contents of this video includes introduction about analytical centrifugation, principle of analytical centrifugation, steps of analytical centrifugation, applications of analytical centrifugation, advantages of analytical centrifugation, and limitations of analytical centrifugation. Analytical centrifugation. Analytical centrifugation is a separation method where the particles in a sample are separated on the basis of their density and the centrifugal force. Analytical centrifugation, the rate at which the particles sediment is influenced by their size, shape, and density. Heavier particles sediment faster, while larger particles sediment more slowly. By monitoring the sedimentation process over time, valuable information about the particles' properties and behavior can be obtained. Analytical ultracentrifugation. Analytical ultracentrifugation, or AUC, is one of the most powerful methods for the determination of molecular mass, shape, and interaction of macromolecules and their complexes in solution. The first analytical ultracentrifuge was developed by the Swedish chemist Theodor Svedberg together with J. Burton Nichols and co-workers in Uppsala, Sweden during the 1920s. Principle of analytical centrifugation. Analytical centrifugation is based on the principle that particles that are denser than others settle down faster. Similarly, the large molecules move more quickly in the centrifugal force than the smaller ones. Analytical ultracentrifugation for the determination of the relative molecular mass of a macromolecule can be performed by sedimentation velocity approach or sedimentation equilibrium methodology. The hydrodynamic properties of macromolecules are described by the sedimentation coefficients. They can be determined from the rate that a concentration boundary of the particular biomolecules moves in the gravitational field. The sedimentation coefficient can be used to characterize changes in the size and shape of macromolecules with changing experimental conditions. Three optical systems includes absorbance, interference, and fluorescence are available for the analytical ultracentrifuge that permit precise and selective observations of sedimentation in real time. Steps of analytical centrifugation. Step 1 is sample preparation. Small sample sizes, typically ranging from 20 to 120 mm3, are collected and prepared for analysis. These samples contain the biomolecules of interest and are placed in specialized analytical cells or tubes. Step 2 is loading the analytical cells. The prepared samples are loaded into the analytical cells, which are designed to withstand the high speeds and forces generated during centrifugation. These cells are then carefully positioned inside the ultracentrifuge rotor. Step 3 is centrifugation. The ultracentrifuge is operated to generate a centrifugal force that causes the biomolecules in the sample to migrate through the solvent. This migration occurs radially outwards from the center of rotation. The centrifugation is performed at high speeds, typically in a range of tens of thousands to hundreds of thousands of revolutions per minute. Step 4 is optical observation. During centrifugation, the position of the biomolecules relative to the center of rotation is determined using an optical system such as the Saranraj optical system. The Saranraj optical system allows for the visualization and measurement of the concentration gradient and movement of the solutes in the sample. Step 5 is data collection. As the biomolecules migrate radially, the distance from the center of rotation is recorded or monitored using the optical system. The data obtained includes the solute concentration and the square radial distance from the center. Step 6 is analysis and determination. Based on the data collected, a graph is constructed plotting the solute concentration against the square radial distance from the center of the rotation. The graph, known as a concentration gradient profile, provides valuable information about the molecular mass and distribution of the biomolecules in the sample. The final step and the step 7 is molecular mass determination. By analyzing the concentration gradient profile, the molecular mass of the biomolecule can be determined. This is achieved by comparing the experimental data with theoretical models or reference standards. The relationship between the solute concentration and the squared radial distance helps in calculating the molecular mass. Applications of analytical centrifugation. The first application is purity determination. Analytical centrifugation can be utilized to assess the purity of macromolecules. By analyzing the sedimentation behavior of a sample, it is possible to identify and quantify impurities or contaminants present in the macromolecule preparation. This information is crucial for evaluating the quality and integrity of the sample. The second application is examination of molecular mass changes. Analytical centrifugation allows for the examination of changes in the molecular mass of supramolecular complexes. Supramolecular complexes are formed when multiple molecules interact to create a higher order structure. Through centrifugation, researchers can monitor the sedimentation behavior of these complexes and determine if any changes occur in their molecular mass under different conditions or in response to specific stimuli. The third application is determination of relative molecular mass. One of the primary applications of analytical centrifugation is the determination of the relative molecular mass of solutes in their native state. By monitoring the sedimentation behavior of macromolecules in solution, the relative molecular mass can be calculated based on the sedimentation coefficient. This information provides insights into the size, shape, and composition of the macromolecules and helping researchers to understand their structure and function relationships. The fourth application is characterization of macromolecular interactions. Analytical centrifugation enables the study of macromolecular interaction. By analyzing the sedimentation behavior of a sample containing interacting molecules, researchers can gain insights into the stoichiometry, affinity, and kinetics of these interactions. This information is crucial for understanding the mechanisms of molecular recognition, complex formation, and functional associations. The fifth and final application is assessment of protein conformational changes. Analytical centrifugation can also be employed to examine conformational changes in proteins. By subjecting proteins to different experimental conditions such as changes in temperature, pH, or ligand binding, the sedimentation behavior of the protein can be monitored. These observations provide valuable information about the conformational transitions and structural changes in the protein molecule. Advantages of analytical centrifugation. The first advantage is high resolution. Analytical centrifugation allows for the measurement of the size, shape, and density of particles with a high degree of resolution. The second advantage is versatility. Analytical centrifugation can be used to measure a wide range of particles, including proteins, nucleic acids, cells, and organelles. The third advantage is non-invasive. Many analytical centrifugation techniques are non-invasive, meaning they do not alter or damage the samples during the measuring process. The fourth advantage is high sensitivity. Analytical centrifugation techniques are highly sensitive and can detect even small amount of samples. Limitations of analytical centrifugation. The first limitation is complexity. Analytical centrifugation techniques can be complex and require specialized equipment and expertise. The second limitation is limited to measuring particles based on size, shape, and density. Analytical centrifugation is limited to measuring particles based on their size, shape, and density, and it may not be suitable for measuring other physical properties. The third limitation is time consuming. Analytical centrifugation techniques can be time consuming, particularly if multiple measurements are required. The fourth and final limitation is expense. Analytical centrifugation techniques require specialized equipment and consumables which can be expensive. Dear viewers, that's all about the analytical centrifugation. Thank you for your support. Thank you.